Welcome to today's panel on chemistry, a tool to understand human health. I am Juan de Pablo. I am the Vice President for Innovation, Science Strategy, National Laboratories, and Global Initiatives at the University of Chicago, and I am also a professor of molecular engineering at the Pritzker School of Molecular Engineering. Before we begin, a few housekeeping items. To listen to today's program in Mandarin, click on the interpretation icon at the bottom of your screen and select Chinese. And please feel free to use the Q&A function to submit questions in either Chinese or English throughout the event. Modern science, technology, innovation, they all require collaboration and partnership at scale to take on global challenges and provide tangible solutions. To work at the highest levels, no institution can do it all. It is through intentional partnership that we can have the most impact on science and ultimately on the world. To increase our impact, we leverage cross-disciplinary collaborations, our relationship with two Department of Energy laboratories, partnerships with industry, an innovation ecosystem, and a network of inquiry that extends to our global science partners. The scientific community's response to COVID-19 is a very good example of what can happen through collaboration. Chemical biology is a great example of a field that is generating new knowledge about fundamental science while also creating translational applications that are already reaching the marketplace to change clinical practice. This integrated field requires a collaborative approach between chemists, physicians, biologists, computer scientists, engineers, and more. We will hear more about this exciting area of research today. Professor Chen is chair of PKU's Department of Chemical Biology. The focus of his research is to develop novel technologies for protein manipulation in living cells, which include protein-based bioorthogonal chemistry and precise protein engineering. The goal of his research is to utilize the valuable tools developed to address fundamental questions in the life sciences. He obtained a bachelor's degree in chemistry at Peking University and a PhD in chemistry with Professor Chuan here at the University of Chicago. Professor Chen studies glycosylation in protein quality control and immunology. His research takes a highly interdisciplinary approach to tackle important questions related to human health and disease, using the techniques of chemistry, biology, and nanoscience as tools to study and manipulate glycosylation in living systems. He is also the dean at the College of Chemistry and Molecular Engineering. He completed his bachelor's degree in chemistry at Tsinghua University and his PhD in chemistry from the University of California at Berkeley. Chuan is a John T. Wilson Distinguished Service Professor at the University of Chicago and is an expert in the field of RNA modification biology and cancer epigenetics, which is the study of how behavior and environment can cause changes that affect the way that genes work. Chuan was the first to champion the idea that modifications to RNA are reversible and can control gene expression. Control of RNA, the molecule that carries DNA's message to the protein-making machinery of the cell, can affect the outcomes of gene expression. Chuan received his bachelor's from the University of Science and Technology of China and his PhD in chemistry from the Massachusetts Institute of Technology. Chuan, it would be great if you could provide some context for today's discussion. Can you briefly explain to the audience what chemical biology is, why it is relevant to human health, as well as your own partnership with PKU and how it has contributed to the advancement of your own work?
Yeah, great. Thanks, Juan, for that great introduction. if we want to really understand the chemical biology, I guess many of you actually know biochemistry, right? What is biochemistry? Well, biochemistry is really one's trying to understand the chemistry of a biological entity. It can be a protein or RNA, right? The exact chemical function or mechanism of that biological entity. Now, chemical biology is really the other side of the coin. It's really a way to apply chemical tools or chemical methods or small molecules synthesized by chemists to understand the biological processes, to understand the biological network, right? That's actually one aspect of chemical biology. Another aspect of chemical biology, which I always try to emphasize, is also we're trying to apply chemical knowledge to understand the biology. Now, I'll probably just use my own work to give you some examples, as Juan just suggested. My lab used to work on DNA modifications for the last 10 years, right? We probably know DNA has something like 3 billion base pairs, and human being has trillions of cells, but our DNA sequences will be the same in any individual. However, every cell is different. What's going on is DNA carries these modifications, right? In principle, every tissue cell type on the DNA sequence will be the same, but the modification patterns are very different. So my laboratory developed the tools to try to map these modifications on DNA in order to understand the complexity or the epigenetic landscape of each individual cells. Right? If you understand that, and the cancer cells versus healthy cells, you start to understand how cancer cells grow out of control. Now, in one particular example, we apply a synthetic molecule with an enzyme. We can label a particular DNA modification with a chemical tag that allows us to enrich the modified DNA, that we can subject that to a next generation of sequencing. So this allows us to map all the modified DNA fragments with this new sort of a chemical method. Now that leads to really a enabling tool, allow biologists to understand, first to detect the modifications in different cellular contexts, understand the biology behind these modifications. Now, in recent years, this method, because it works robustly and works with small amount of materials, we've further advanced this to study so-called liquid biopsy, right? You can take blood, 3 to 4 mil, isolate, we call it circulating cell-free DNA, so the tiny amount of DNA released from almost every tissue into your bloodstream, they're so tiny, very small amount. However, if an individual carries cancer or other diseases, there's small portions of these DNAs actually come from the diseased tissue. Apply our sensitive, robust method allows us to capture these cell-free DNAs for early diagnosis, but also prognosis of diseases. So this is just one example of sort of applying chemistry to understand the biology and further advance this to potential patient care, right? As you'll hear from Professor Peng Chen, Xing Chen, and many others working in chemical biology, were constantly synthesizing and creating new molecules, new methods that allow us to probe biology. That's really one of the key aspect of chemical biology. The other side of chemical biology, as I always say, is also our knowledge in chemistry, which has been extremely important for understanding biology. As Juan said, my lab has been really working on RNA modifications or chemical modifications on RNA, which was discovered almost half a century ago. But it was really work from our lab started to re-spark the extensive interest of this field in biology. I think a major reason is because of our chemical knowledge, right? we apply about 90–95% of our lab applied standard biological approach or biological tools. However, that 5 to 10% of chemistry, the way we think about problems chemically while also biologically, really help us to establish the importance of these functional marks on RNA and think about what they imply and how can these chemical marks lead to, and identify new proteins, enzymes that manipulate on these marks, and some of these become targets for, say, anti-cancer therapy research. So it's an exciting time to become a chemical biologist. There's really an interdisciplinary research. We all apply chemistry, however, every single one of them study very different biological questions. As I always said, the chemical biologist, we have to reinvent ourselves every 5 years. We reinvent new tools, but also go into different biological areas to make new discoveries, to impact, hopefully, therapeutics and medicine. University of Chicago has a long history of interdisciplinary research. We also have a long history with relationships with China. It's dated back for a long, long time. Many names the audience in China very familiar with, right? Dr. C.N. Yang Zhenlin, Dr. T.D. Li, Li Zhengdao, they both did their received their graduate training from the University of Chicago, right? In chemistry, there are also many pioneers in chemistry. In Chinese chemical community received their training from University of Chicago as well. Example is perhaps some of you know of is Professor Liu Youcheng. Is really the pioneer of radical chemistry and bioorganic chemistry in China, and he received his postdoc training. So that tradition, of course, lasted for many, many decades. Here we have Professor Chen, and I know at Peking University there are a number of faculties who were trained here in University of Chicago. In summer, we started a chemical biology seminar series in which we have a faculty from University of Chicago, another from Peking University, each present 20 minutes, and that was a great sort of event, really covered broad scopes of chemical biology, and I was very delighted to see that all the faculties showed their best work, and really the diversity of the research subject went way beyond sort of a traditional chemistry or just a biology, and it was really a spectacular show of modern chemical biology research. With that, I'm delighted to be involved in this, and I think there are just a lot of opportunities for us to explore in chemical biology, but also a lot of opportunities for us to really join the force between scientists in China and then scientists in US, particularly in Chicago, to try to accomplish our common goals. Thank you.
Thank you, Chuan. That was very interesting. Peng, as chair of the Department of Chemical Biology at PKU, I would be interested to learn about some of the recent advances in translation and therapeutics. Also, as a graduate of UChicago, I would also be interested in your perspective on the importance of engagement between the US and China.
Hi, sure. Yeah, I will. I have some slides to share. Thanks, Juan, and hi, everybody from both Chicago and Beijing. It's my great pleasure to join this exciting event. And I just made some slides to deliver more message with this to celebrate this exciting 10th anniversary event for the University of Chicago Beijing Center. I will show you how PKU and ULC are joint force and the interface of chemistry and biology to propel the frontier and this interface and to benefit human health. And I'm a professor and chair of chemical biology, and I received my PhD in chemistry at the University of Chicago in 2007. Our department was founded back in 2001, which is one of the earliest departments founded in China, and also actually one of the earliest chemical biology department in the world. And in 2011, we founded another chemical biology joint center, which has been led by Professor Chen and the University of Chicago. And the name is called the Synthetic and Functional Biomolecule Center, SFBC for short. Right now, this department has 7 full-time PIs. 2 of them, including myself and Dr. Guifang Jia, the lady in our department, actually both graduated from Chicago with, I received a PhD and Guifang had her postdoc training with Chuan. And also we have a few associate professor and co-PIs, and Professor Chen now is our guest professor. Our research is that the interface of the chemistry and the biology can fall into 3 major parts. One is the chemical modulation and manipulation of biomolecules. The second direction is the chemical and the genetics and the proteomics. And the third direction is the biophysical chemistry. So we develop chemical tools to elucidate the biological mechanism. And we also develop chemical labeling and functional manipulation approaches on biomolecules. We also are interested in chemical biology-driven clinical and medical research. And also we have people trained in biophysical background, so they work on biophysical chemistry and computational biology. So together we hope we are aiming to build a world-class interdisciplinary research program and the interface of chemistry and the life sciences. And we, of course, beyond PKU, we have lots of collaborations with other institutes, particularly with PSD and the chemistry department at Chicago. So we hope to build a world-leading research center and apply chemistry tools to and the chemistry knowledge to explore fundamental biology questions, which will ultimately lead us to a deeper understanding of various life processes, as well as human diseases. This will not only provide a new technology and research strategy, but also will deepen our understanding to address challenges in both fundamental biology and in human health. But due to the time limit, I will not go through the research details, but I would show 2 slides with the ongoing clinical translational science that is actually going on in our department center, and also the joint force between us and UChicago. One is this 5 HMC-centered epigenetic platform for early cancer diagnostics and drug discovery. This original 5 HMC sequencing and capturing technique was developed and pioneered by Professor Chen at the University of Chicago, but later was applied to clinical relevant research in China. As you can know, we have a larger population and patients. So actually this flow has been this workflow has been established in China, where the companies start to collect samples from patients and then apply this sequencing strategy for early diagnosis of various cancers, such as liver cancer. And then this will guide us for more target drug identification, and then we'll screen for drug target. So this is a whole joint efforts between China and US. So I really echo what Guang just mentioned, that this is really beyond the science, scientific discipline, and beyond the the country. It's really an international effort to drive this frontier of cancer diagnosis. The second direction is that we are developing single domain antibody-based therapy for immunotherapy, particularly for solid tumors, because we know that although the antibody-based therapy has really one of the major driving force to treat all kinds of diseases, but there are lots of diseases are not curable by the current antibody technology. One key issue, particularly for solid tumor, is the large size and the low penetration of the full-length antibody. So we are actually about 5 or 6 years ago, we started to build this we call nanobody or single-chained, single-domain antibody platform, where the antibody scaffold is only about 1/10 the size of the full-length antibody, which bears lots of advantages. And these antibodies were originally came from camel and shark, but we can now manage to make the whole high throughput discovery and the platform built in our department and center. And as you can imagine, due to the small size, this has a better penetration. And then we are using all kinds of chemistry technology to make a further multivalent conjugation antibody. And then this has also been pursuing on a clinical relevant setting. This is a joint effort with a few PIs, and also Chen, Professor Chen, he gave lots of advice on this exciting direction. And then we also have many research activities, such as the annual symposium of SFBC back in 2011, and in 2013, actually, is IBD. All the faculty from IBD and UChicago came to visit PKU campus. I'm sure you see lots of familiar faces, and they are visiting us and give an exciting symposium. And also we have a summer camp, and there's a joint series of PKU and UChicago seminar series that Chuan just mentioned that last summer is very successful, and we're hoping to having this in the near future. And we are the first department to have the chemical biology lab course in China. Actually, we are training the next generation of undergraduate students who are actually they can expose the advanced chemical biology lab and their junior or the senior years. And we are actually publishing lots of textbooks on the training guidebooks in both in Chinese, and we're actually working on the English version textbook very recently. So last but not least, how the UChicago training impacted me, myself, and also many scientists that graduated or received training from University of Chicago. One of the I would say one of the representative work is that we, thanks to all the colleagues from UChicago, we established this PKU and UChicago cooperative education research program about 5 years ago. And every summer there will be about 6 undergraduate students from chemistry to do exchange program at the U of C lab. And this has been nearly about 30 students in the past 5 years has received this training program. And I believe some of them are now still at the UChicago pursuing their PhD degree. And also, we are actually very excited to let you know that we're assigning the second phase, renewing this agreement. So we will have another exciting 5 years to have this program, have more people get interdisciplinary and joint training. And so, yeah, this is really Chuan and Kate puts lots of efforts on this. And we're now about to sign the agreement. And it's a really exciting opportunity. And I learned that is beyond the PSD, the BSD are doing the similar program, even maybe hospital and material science division, they also are working on similar programs. So this is really one of the representative work between the two universities. So let me summarize that. I would see, the joint collaboration efforts between two universities or even between two countries, really pushing the frontier of not only basic science but also clinical science and the training and education of next generation scientists. So really, I'm really looking forward to our more fruitful collaborations. And finally, again, thank you all, and happy birthday to the Chicago Beijing Center.
Thank you very much, Peng. I also look forward to more collaborations with PKU, certainly in the area of chemical biology. Let me now ask a question of Xing Chen. Xing, I understand that all three of you collaborate extensively through the Synthetic and Functional Biomolecules Center at PKU. Can you tell us more about this initiative, please?
Of course, Juan. And also, I'd like to join Chuan and Peng to say that I'm glad to be part of this wonderful event. As we all know, Peking University and UChicago has collaborated for a long history. And part of this partnership between these two universities, I think Synthetic and Functional Biomolecules Center, SFBC, which Juan just mentioned, it has been playing an essential part for fostering cooperation between the two universities. SFBC was founded in 2011 at Peking University as an initiative to foster research at the interface between chemistry and life sciences. The center has now have about 10 research groups. They are coming from different colleges and departments, including our college, College of Chemistry, School of Life Sciences, and also PIs from medical school. For the past decade, research at SFBC focuses on epigenetics, RNA modifications, protein chemistry, chemical biology, chemical proteomics, and chemical genetics. The goal of the center is to build a world-class interdisciplinary research center in chemical biology. We hope to provide new chemical tools for studying important biological questions through the integration of chemistry, biology, and medicine. In the long run, we hope to contribute to solve fundamental issues related to human diseases. And I think I have to mention that Professor Chen has been the director of SFBC. Chen, although a little bit sadly, is stepping down this year upon completing his directorship. He has made tremendous contributions to this center and chemical biology research broadly at Peking University. Chuan helps to recruit talented people to the center, including myself and Peng, design the research directions. In SFBC, Chuan also talks to young faculties, giving research and career development advices. Without Chuan, I think the center could not be as successful today. The center, as I just said, also serves as an important node in a partnership between PKU and UChicago. For example, we organized joint conferences. SFBC has worked closely with Chicago Center here for many events. There are many other examples, some of which Peng and Chuan have mentioned earlier. I think it is fair to say this has been a model of successful international collaboration. And coming back to the research part of SFBC, I think the center for the past decade has developed many chemical tools to study important biological processes and biological questions. And for the next decade, I think Chuan and the PIs in the center, we have been discussing what should we do for the next decade. And we came to a very consensus a plan that we should push forward the direction towards the clinical and translational research. And one of the reasons, or one of our advantages, is that Peking University has 8 affiliated hospitals, and very good ones. I think we are in the good position to push forward or to apply the chemical tools or chemical biology tools we have developed here for the past decade in the center. So we plan to focus this direction for the coming decades in the center for the new research direction. This, I think, is also a mutual interest between Peking University and UChicago. Speaking of international collaborations, at the college level, we also believe it is an essential component for pushing forward the boundary of the science research. I think here's just a very interesting, or a very typical number I just got last week. If we do a statistical analysis on the publications of the whole college, we found that the percentage of the international cooperation works has increased. It has been increasing for the past several years. And I also think the international cooperation should be driven by, mainly by the research interest, as I just mentioned, pushing forward the chemical biology frontiers towards clinical and translational research has been a mutual interest between Peking University and Chicago, I believe, as well as other major universities and institutes. We hope to work together to push forward the frontier of chemical biology. I think it is important to keep an open and cooperative mind. Finally, with the very successful platforms, including SFBC, Chicago Center, and many exchange programs, I'm sure we are looking forward to continuous, deeper, comprehensive collaborations between University of Chicago and Peking University. Thanks, Juan.
Thank you very much, Xing. That was very helpful. I think that we'd like to move on to the question and answer portion of our panel. The questions have been coming in, and I'd like to start with one that we all care deeply about, and that pertains to a comment that Chuan made earlier. He said that chemical biologists have to reinvent themselves every 5 years. And that is so true. And the question really to all of you is, how do you train people in academic programs that are so flexible and nimble and courageous to be able to transition from one discipline or one problem to another every 5 years? How do you do that? Perhaps, Chuan, since you said it first, I'll ask you first.
Yeah, that's actually a great question. I think that's also the sort of the philosophy here at the University of Chicago, in a way that, we I always think as a PI, we train students to be an independent thinker, right? There are two things I always say we train them. One, sort of provide them with basic skills, right? They can really learn fundamental, so sort of basic chemistry, chemical biology, molecular biology, biochemistry skills. So they can apply to different disciplines, but most importantly, we want to train them as an independent thinker, right? So in my lab, for instance, I always give students two projects. At the beginning, they will follow a senior student, postdocs, right? Learn how to think through science. And about second, third year, they take on very independent projects in a way to become, fully independent in 2 years. By the time they graduate, write their thesis, hopefully they're independent thinker, and then they can apply their tools to study other disciplines. So I think that's sort of the way we want to train our students. But I will have ask the other two Chens to comment.
Yes, Peng, perhaps you can tell us a little bit about what traits you look for in students to be able to transition quickly.
So I will add.
Go ahead.
Yeah, I will add on top of Chan's comments that I think this reinventing thing is are actually in two folds. One is the motivation, the curiosity driven by scientists, and the other is really the clinical needs. We are constantly been challenged and facing challenges in the the human health. For example, the COVID-19 is really a pandemic and a global issue right now, and we as a scientists need to address these issues with all kinds of discipline. It's really joint efforts. Actually, I saw on the questionnaire there are people also asking Chuan's comments about how his RNA research will going to impact the study or the treatment of COVID-19. I will leave that to Chuan, but I just want to mention that our nanobody platform we also may been utilized to treating or to preventing these the virus. Particularly, we are seeing some new mutant strains of the virus that are really mutated so fast they may escape even the vaccine. So the neutralizing antibodies, the next generation neutralizing antibody, I would say would be something that need to be further developed by not only on the clinical, but also the basic research breakthrough will benefit those frontiers. That's my comments.
But Xing, do you have any comments on this?
Yeah, I just like to add one point. I think for reinventing chemical biology, I think the students need to be motivated or self-motivated.
Yeah, that's certainly a very important quality. You mentioned COVID and RNA, and I think, yes, the COVID vaccines have been one of the great triumphs of RNA research in recent years. With the development of these vaccines, of course, a question that arises is how have your work or that of your partners, Juan, contributed to the development of the vaccines? Can you put that in context?
Well, actually, the RNA vaccines, the basic biology and the discoveries for RNA vaccines being out there for almost 2 decades. I think scientists have been thinking about using RNA as vaccines for a long time, but there are major problems, major problems. They induce immune response, quite dramatic immune response. So that's not very good. So there are 2 things people solved that. One is they figured out a way to purify RNA. So it turned out impurities induce immune response. The second way is exactly the type of thing we work on. Is they realized that when they add modifications, so it turned out whatever RNA vaccines you take, they're not native RNAs, right? Every single uracil. So we know there's AGCU. Every uracil is replaced by a modified uracil called 1-methyl-pseudo-U. It turns out when you have that modification, you pretty much prevent all the potential harmful immune response. That turned out to be a really turning point for applying RNA vaccines in real human cases. So we've been quite interested in modification biology and how does RNA modifications really communicate with innate immunity. I would say that since pandemics, we've been basically screening for a variety of different RNA modification systems. There's actually 170 different RNA modifications existing in nature. So we screened a bunch of RNA modification systems, and we realized that a number of them actually involved in antiviral immune response. So we're actually going through those targets and trying to figure out if they're useful for viral prevention or treatment or even cancer immunology. This is a very good example that something people studied for a long time. People don't really think it's that important, it's useful, but suddenly turned out to be extremely important for today's COVID vaccine, right? It's basically a study 15, 20 years ago. People realized that these modifications were preventing innate immunity.
Yeah, a lesson for all of us: the fruits of fundamental research are always useful in unexpected ways. And this is a good example of that. Building on your answer, and really this is a question for all of you, perhaps Peng, you want to take this one. There's increased concern that new mutants of the virus are coming up. And one of the questions from the audience is, can we win this war against this virus as new mutations come out apparently very rapidly? What are your thoughts on that? And how can fundamental research help us move faster in that direction?
Okay, yeah, that's really a challenge, and I'm not an expert in the virology, but yeah, I will try to answer. Yeah, we are seeing really the increasing mutation burden from all over the world, and I would say there are two folds to prevent this. One is that people have to wear masks, have to do the physical protection. Even the sort of the lockdown policy in China, actually, it really works. So I would say before we are getting a really solid conquer over the virus with our technology, everybody has to be, self protection, like quarantine by, that turned to be the most effective way in many different countries. Yeah. Secondly, of course, we need to finally conquer this virus with our technology. I'm convinced that we will be able to do this. For example, another U of C alumni, Professor Sunny Xie, who did postdoc at UChicago and later a Harvard professor, and I'm sure many people know him, and he's returned to China about a few years ago, and after the outbreak of this pandemic, he devoted himself all the way to do the neutralizing antibody research. And as far as I know, yeah, his company, the company is moving really fast to making the neutralizing antibody. I will say we'll eventually conquer this with the advanced technology ranging all the way from single-cell sequencing and high-throughput sequencing to RNA epigenetics and to antibody engineering. And also, we need all the efforts from clinicians. It's really a joint effort. You may not know exactly what basic research will finally make this happen, but as just Chuan mentioned, the RNA vaccine has to be a really fantasy a few years ago, but now it's really everybody's taking the RNA vaccine actually. So yeah, this show how the power of the basic research will impact. And obviously, I think that's the way to go. And maybe Xing has something to share.
Xing, what are your thoughts on this?
I don't have too much to add, but I just to echo what Peng just said. I think the scientists all over the world, whether or not they are in the field of virology, if anyone would be happy to or do his best to devote into this research. I think this is the the hope or the driving force to conquer this virus.
Yeah, one of the important relationships that the development of the vaccines has highlighted is how critical it is that we have strong partnerships with industry. Speed is of the essence. What we do in the lab has to be translated rapidly and scaled up. With that in mind, in what ways do you think academic institutions should be engaging with industry to advance translation, certainly in the area of therapeutics, more effectively? And do you have examples of how those partnerships should be that you can share with us?
Yeah, I think I can start with some things we have done here at in our College of Chemistry at PKU. I think different countries or different universities have different stages or situations in terms of translational research. So I think like several years ago, we evaluated how what should be the best or one of the best or things we should try. So one thing we have done is that we sort of established a organized translational research platform. So our thinking is that the market or the industry would invent a very mature research results, and there is a gap between very basic research in labs and the sort of mature results that the industry or the market will take on. So so our efforts to bridging this gap is to organize a platform which the faculties when they are interested in push forward their technologies, tools, or results towards that direction, we will provide some fundings and we'll connect them to industry for a very early stage of development. We have built such a platform not only for chemical biology, but also for different directions in chemistry, including energy, materials, and we hope through that platform we can connect basic research and translational more efficiently.
Very interesting. Chuan, perhaps you can offer your own US perspective on how that would be done and contrast what is taking place here at UChicago with what is taking place in China.
Right. I think if you look at least in drug discovery historically, right, 20, 30 years ago, these big pharmaceutical companies doing a lot of outstanding research. But that trend has slowly changed, that they start to rely on biotechs, right? The biotechs, small biotechs take on the high-risk projects and then develop those projects or ideas, and then they get bought, acquired by big pharmaceutical companies or team up. So it becomes sort of the small biotechs with professors involved are sort of moving the innovation from. This is the reason why Boston, Bay Area, San Diego, right? When you have a top-notch research institutes, best minds in academia, they come out to start biotechs. But as we also witnessed that the incubation has been getting closer to the lab itself in many ways, because at the end of the day, the largest value, if you're industry people, you always talk about the build the value. The biggest value comes from innovation, right? A fundamental basic discovery or breakthrough that brings in the real value, which makes you very distinct, unique. And often that comes from research lab. So I would predict, we started to see this, that a lot more sort of first stage translational work started to go into individual laboratories with industry partnership or sponsorship. I think we're going to see a lot more in the next 5-10 years. And it's important for University of Chicago, other institutes to adapt to that change here in the US. In China, I think if you look at places like Suzhou and Shanghai, it's just like Boston. There's a lot of innovation going on with biotech. But I think the universities there, as Professor Xing Chen just said, is also trying to catch up, or trying to really jumpstart this efforts to develop innovation directly in PI's laboratories.
Yeah, I see what you're saying, and I have a related question now, which is relevant for all of you, and that is how do we reward translational research as opposed to fundamental research? Our traditional model has been one in which you do fundamental science and you publish trend-setting papers in some of the finals journals, but that doesn't necessarily translate into impact. I think impact can be discovering a great new drug or therapy and taking it all the way from the laboratory to a company and to a drug that's used by millions. How do you reward that type of activity, both in China and in the US, so that we can have more of that?
Maybe our Chinese colleagues can go first.
I'd like to hear, so perhaps Peng, you're chair of your department at PKU. How do you encourage people to be more entrepreneurial?
Yeah, we actually, in the past few years, we have constant chat with Chuan and Xing. Well, first, we are trying to train the next generation young top talent to be more geared towards the translational research rather than pure academic. The academic, first of all, the position will eventually be filled, right? There will be much space to explore in those translational area. And I would say that no matter which direction, the top talents is always a key issue to success. But for as you mentioned, how to reward, I will see the market, the patients themselves are the rewarding. If you make your drug or your diagnostic really all the way to clinical, you not only make impact, but you are make yourself rich, right? These are all the rewarding eventually. I know it takes a hard way, but just as scientists publishing papers, it does not equal to an impact. They are building all these impacts. You have to hold the faith that it will eventually be converted to basic breakthrough in basic science, but also really a translational. We need to make examples, and China is making good examples, and the people are following him. So, yeah, we are testing it, but I think, you brought a great question, and we're on the way to try and test error. Yeah.
Chuan, anything to add?
Yeah, actually, Well, that's a great question, because in our academic evaluation system, how do we evaluate that, right? If we have somebody, it may take 10 years for the, efforts to pan out. Do we give them a tenure or not, right? Because there's no clear academic accomplishment. I think that's a great question. I don't know if I have an answer, but I think one thing I learned in my own experience is that I hate to say this, but money is very fair in a way. I realized the capital is actually very fair, because they're trying to get more sort of money in a way. They always invest on something new and promising. And so one sort of parameter for me to look at the translational efforts in US, at least, I kind of look at the situation and ask how much money is being invested. There are so many venture capitalists spending so much time trying to analyze every bit details of everything possible, right? I think I joked that money is actually a better evaluator than often us, because, but you can't use that as an academic parameter. So I'm with you. I actually don't know. I think a more tolerated environment might be the way to get there. Like mRNA vaccine, the ones who did this, they never had the sort of recognition they deserved until this year, right? So,
absolutely. So
I would say one last word. I think it really depends on how unique somebody is trying to do something very unique. They have their own niche. Doesn't matter that's basic or translational. I think that's probably something we should always value.
I think so. I think uniqueness, distinctiveness, and again, a balance between our traditional scholarly endeavors and entrepreneurial activities that will impact society. I think finding that balance is critical. And on that note, I would like to thank you all again for participating in this panel. I would like to thank our audience for staying with us throughout this presentation. I hope you found the discussion as interesting and invigorating as I did. Thank you very much. And I'd like to remind you that we have the last in this series of events next Thursday at 7 p.m. Central Time. I hope that you will join us for a fireside chat with President Zimmer himself. Thank you very much.